Hello, everybody. Good evening, all around. Good evening, everyone. How are you all doing? Am I audible? Can we have some quick highs and good evenings around so that I know you people are able to hear me? Good evening, Amit. Good to see you here. Good evening, Ahisan. Okay, so are you all ready for the question discussions today? So आज हम बात करेंगे म्यूटेशंस पे. अगर आपने पिछले ईयर के एग्जाम पेपर्स देखे होंगे तो यू माइट हैव नोटिस दैट म्यूटेशंस लॉट ऑफ क्वेश्चंस आर आस्क्ड फ्रॉम म्यूटेशंस. Okay, so there will be questions from chromosomal aberrations as well as questions related to gene mutations, especially the segment where we talk about the various mutagens. Okay, कौन सा mutagen है, क्या काम करता है, depending on that there are different kind of questions every session. Almost every session you will see questions coming from that part. Okay, so today we will try to see some specific examples of those questions, and definitely we will try to solve them together. And I will also let you know that will help you in solving those questions in future. Okay, everyone ready? Good evening, Sagrika. How are you? All right, let's start. So what I'll do again, as we do always, so I will turn off my video so to make sure that there is no lag in the video, and then smoothly we will continue with our class. Okay, so very first question, and this question was there in last CSIR exam itself. So is heterozygous for a reciprocal translocation as shown below in the following diagram. The following statements are made about the segregation of such chromosomes during meiosis and gamete formation. So first of all, what is translocation? Translocation is a type of chromosomal aberration where segments will pass from one, there will be an exchange. So translocation is movement of chromosomal segment between non-homologous chromosomes. Okay, so suppose you have one chromosome which has A, B, C, D, E, and you have another chromosome that has M, N, O, P, Q. Okay, so they are two non-homologous chromosomes. How do we know that they are non-homologous? Because there are different gene loci present on them. Okay, now this non-homologous chromosome, so if there is an exchange of genetic material between this non-homologous chromosome, then it will be called as reciprocal translocation. तो अगर इन दोनों के बीच में reciprocal translocation होगा तो कैसा बनेगा? Suppose one part it came, okay, so M N यहाँ आ गया, बाकी पार्ट है C D E. okay, और इसमें क्या हो गया, यहाँ पे A B आ गया. Okay, or OPQ. So this part, it came from one chromosome to here, and this part, it moved from this chromosome to the non-homologous part here. All right, so that will be reciprocal translocation. Now let's see what it is telling here. Good evening, Neetu. Welcome to the session. So this reciprocal translocation has happened and the diagram is given to you. Okay, so basically here what the numbers are meaning. So 1 and 2 are two different chromosomes. Okay, so out of one of that has participated in translocation with one member of 2, and these are T1 and T2. Okay, so अगर हम इसको ऐसे लिखेंगे तो ये कैसा बनेगा कि ये क्रोमोसोम नंबर 1 है, ये क्रोमोसोम नंबर 2 है. ठीक है, अब क्रोमोसोम नंबर 1 में एक N1 है. ओके, जो कि नॉर्मल है, जिसमें सारा सेगमेंट सेम रखा हुआ है. ओके, देयर इज नो चेंज हियर. और यहाँ पे T1 है, which participated in reciprocal crossover with, or sorry, not crossover, reciprocal translocation. It participated in reciprocal translocation with another member. Okay, another non-homologous chromosome. So वो है chromosome number 2. ठीक है. So the member of this chromosome number 2 which participated in reciprocal translocation, that we are mentioning as T2, और एक है N2 जो कि normal है, जिसने translocation नहीं किया. Okay, now the chromosomes are written here. So T1, T2, okay, and N1, N2, right? Now what will happen during gamete formation and meiosis? So so about that, some five statements are given. Yeah, good evening, Priyadarshini. So first statement says that the complexly paired 4 chromosome fail to segregate, pass into one cell at anaphase 1, and the cell eventually dies. Chromosome pairs between regions of maximum homology, keeping the translocated part unpaired, and a normal meiosis occurs. Statement C says that one of the way of chromosome segregate is by alternate segregation, okay, where N1, N2 move to one pole and T1, T2 moving to other pole. Alternate segregation produces non-viable gametes. And statement E says reciprocal translocation are considered as crossover suppressors, as no gamete with crossover products are produced. Now select the option from the following that describes the meiotic consequence of such translocations correctly. Okay. Now what happens? What will be the consequence if this kind of chromosomes will enter the meiosis after undergoing reciprocal translocation? Okay, so what will happen is that, see, this T1 and T2, what happens? It has some part which is same as N1 and some part that is resembling N2. Okay, similarly for T2 as well, it has some part that is similar to N2 and some part that is similar to N1. Okay, so these chromosomes, they will form a cross-like structure. This kind of structure will be formed. Okay, so okay, let me show you with the color, then it will be easier for you to understand. So if it is red, so this is N1, okay, and part of this is similar to N1, this part similar to N1. Then this is N2. Okay, this entire one is N2 and this is T2. Okay, so what will happen is that this is N1, this is T1, okay, this is N2 and this is T2. Okay, so T1 and T2, since they have region which is similar or it has homology with both the chromosomes N1 and N2, so they will form a cross-like structure like this. Okay, अब ये वाला जो cross like structure जो form हो गया है, now there are different ways in which it can get segregated. So it can get segregated adjacently, it can adjacent 1, adjacent 2, and there is alternate segregation. Now it is only in alternate segregation होगा कि N1 और N2 एक pole की तरफ move हो जाएगा and T1 and T2 they will move towards one pole. So N1, N2 goes to one pole, T1, T2 goes to one pole. Okay, now these two gametes will be viable. Why? Because all the genes are present in them. Okay, there is no deletion, there is no duplication. Whereas if we see adjacent 1 or adjacent 2 segregation, then it will result into duplication of genes. Okay, deletion and duplication of genes, and because of that, the gametes will be inviable. So there is only one method by which viable gametes can be formed in this situation. It is through alternate segregation only. Okay, so out of all these statements, only statement number C is correct. Okay, so answer will be option 2, C only. Now moving to next question. Now this is another type of question which is very, very common in CSIR exam. Have you guys noticed it? This kind of question, in one of our biotechnica classes, one student said that, yes, ma'am, that donut shape. So yes, this is the donut shape. So you can talk about this donut shape questions. Have you people noticed this kind of diagram? Because it has come multiple times in CSIR exam. Yes, have you people noticed it? This kind of question, have you ever come across this kind of question in previous year question papers? Yes, noticed, Amit. All right. Okay, now let's see how to solve this kind of question. Okay, so what it is talking about? Now, this is also talking about a chromosomal mutation, a structural chromosomal mutation, which we call as inversion. Okay. So what is inversion? Inversion is a chromosomal aberration where the segment of the chromosome will be inverted by 180-degree and reattaches. Okay. So an individual is having an inversion in heterozygous condition. Heterozygous condition, inversion in heterozygous condition. So what does that mean? Heterozygous will mean that, so each chromosome will have two members, right? So this is one member where the segments are arranged like this. Okay. Now the other member of the chromosome has undergone the inversion. Okay. So what has happened? This CD segment, suppose, has got inverted. Okay. So it will become A, B, then D, C, E. Okay. So heterozygous inversion or inversion in heterozygous condition will mean that only one member of the chromosomal pair has the inversion, the other member does not. Okay. Now, in this scenario, complication arises if this cell has to enter the cell division, the meiosis. Okay. Why? Because during the prophase 1 of meiosis, the crossing over has to happen. Crossing over is the process of exchange of genetic material between between the non-sister chromatids of homologous chromosomes. Okay. Now, for crossing over to happen, the first thing that has to happen is alignment of the chromosomes, okay, by the region of homology. Now, since there is a difference here, there is a difference in the order here, the chromosomes will not align. Okay, and because of that, one of them has to form a cross loop kind of structure. Like if one is like this, the other one has to form a cross loop like this. Okay, then only the alignment is possible. Okay, so I'll just like taking the reference from the diagram that is shown here. So if one of the chromosome is like this, the other one has to happen like this. Okay, then only alignment is possible, and then only crossing over can happen through the inverted segment. Okay, now let's go back to the question. So the region as normal chromosomes on the normal chromosome are marked as A, B, C, D, E, F, G, while the chromosome having inversion has the regions of A, B, C, D, E, F, G only in lowercase. Okay. Now the diagram given below shows pairing of these two homologous chromosomes during meiosis, and the site of crossing over is indicated. Okay. So this is centromere, and this is the site of crossing over. Okay, the X here that is shown is the site of crossing over. Now, the following statements are given to describe the inversion and the consequence of crossing over shown in the above diagram. Okay, now see, when you come in biotechnic class, there also we discuss, and as well as if you have studied genetics from any standard book, there also you will notice that when we talk about inversion as a chromosomal aberration, we also have to study the consequence of inversion during crossing over. Okay, so inversion can be of two types. Can anyone tell me the types of inversion very quickly? Inversion is of two types. What are the two types of inversions? Anyone? Yes. So what are the two types of inversions? Absolutely correct, Amit. Paracentric and pericentric. Okay, paracentric inversion and pericentric inversion. Okay. So paracentric inversion is a type of inversion where the inverted segment do not include centromere. Pericentric inversion is a type of inversion that includes centromere. Okay. Now, the consequence of these inversions, if it is happening in heterozygous condition, and what will be its consequence during crossing over, it will vary depending on whether the inversion is paracentric or pericentric. Okay. So, if this kind of questions are given, so very first thing we have to understand is whether the inversion is paracentric or pericentric. Okay. So this inversion, if you see, so we have to identify where the inversion is happening. So inversion is happening in this CDE segment, right? This is the segment where the inversion is happening. Okay. Now this CDE segment, so inversion is there. Okay. But this CDE segment do not include centromere, right? What does that mean? What kind of inversion it is? This is a paracentric inversion. Absolutely correct, Amit. So paracentric inversion पहले ही पता चल गया, तो ये statement तो गलत हो गया. Statement A, that it is pericentric inversion, it is wrong. Statement, it should be F here. So this statement F became correct, that it is paracentric inversion. Okay. Now, once you have determined that, now you have to think what are the consequence of crossing over occurring through a paracentric inverted segment in heterozygous condition. Okay. So when this happens, okay, so when this crossing over happens here, and ultimately the chromosomes will separate. Okay. So first of all, only two chromosomes are two. Okay. So if we see these two chromosomes here, right, ये दोनों अगर है, तो अगर हम सेल डिवीजन की बात कर रहे हैं, so each chromosome will have a chromatid. Okay. So only these two chromatids are participating in crossing over. This one and this one, they are not participating in crossing over. So they will have, or the gametes which will receive this non-participating chromatids, that will be absolutely normal and viable. Okay. So 50% gametes तो ऐसे ही viable होंगे. Now, जिन gametes में the ones who will receive the recombinant chromatids, okay, this one and this one, the ones which are participating in crossing over, they will be non-viable. Why non-viable? Because when this cross loop structure opens and separates, then it will result in the formation of a it will result in the it will be non-viable, because there will be there will be loss of there will be deletion of the genes. Okay. Why there will be deletion of the genes? Because when it opens in paracentric inversion, it will result in the formation of dicentric chromosome. Okay. Dicentric chromatid. Okay. That means a chromatid with two centers. Now that dicentric chromatid, it will be, so one will be dicentric, one will be acentric. Acentric segment will be lost because it cannot be segregated, because there is no attachment point for the spindle fiber. The dicentric chromatid, there is two attachment point. Okay. And then there will be a tug of war and the chromatid will break. So it will result in the formation of a dicentric bridge. Okay. ऐसे बनेगा कुछ structure. Okay. And then it will abruptly break from the middle. All right. And that will result into deletion of genes. So the recombinant chromatids that the gametes will receive, the recombinant gametes will receive, they will have major deletion of genes, and because of that, the recombinant gametes will be non-viable. Okay. So this statement is correct. That is 50% of the gametes having recombinant chromatids will be non-viable, whereas 50% gametes having the non-recombinant chromatids will survive, the ones which did not participate in the crossing over. Okay. Why it is there? Because it will generate, if paracentric inversion is there, so it will generate dicentric and acentric chromosome following separation of chromosomes after crossing over. Okay. So statement B, E and F are correct. So answer will be option 3. Again, I hope all of you understand, all of you remember, and please note down the doubts. And as you know, at the end of the class, there will be a Zoom link and we will have further discussion, one-on-one doubt clearing session. So you can come up with your doubt and we can discuss it there. Okay. Now, moving on to another popular category of question where we talk about the mutagens. Okay, so this kind of questions also, if you have gone through the previous year question paper, so you might have noticed this kind of questions where the mutagen is given. Okay, this is called as mutation reversal study. Mutation reversal study. Now, this mutation reversal study is done to find out the mutation caused by a particular unknown agent. Okay, so what we do is the unknown agent is used to produce some mutation in the cells, and those mutants are reversed, okay, using some known mutagens. Okay, and from that analysis we try to find out what was the kind of mutation that is induced by the unknown agents. Okay, so in this it is given that a scientist synthesized 4 new chemicals which has mutagenic potential, named them as C1, C2, C3, C4. So this C1, C2, C3, C4 are the unknown mutagenic chemicals. So he tried to analyze the nature of the mutations caused by them and obtain the following result. So mutation produced by mutagen C2 and the mutation reversed by 4 different chemicals were used: 2-aminopurine, nitrous acid, hydroxylamine, and acridine orange. Now, which of the following answer describes the mutagenic potential of the chemical? Now, to answer this question, the trick is to remember what kind of mutation is induced by this specific chemical agents. Okay, so 2-aminopurine, what is it? It's a base analog. So base analog produces what kind of mutation? Transition. Okay, now if base analog is capable of reversing a particular mutation, that means that first mutation, the forward mutation, was also a transition. Okay, so that is how we will correlate. Okay, so 2-aminopurine base analog produces transition. Nitrous acid is a deaminating agent, and that also causes transition. Hydroxylamine is a hydroxylating agent that also causes transition, but only one specific type of transition. Okay, not so. Now, what is transition? Transition is a base substitution mutation where a purine is replaced by another purine and a pyrimidine is replaced by another pyrimidine. So basically, it will be Ta base pair will change into Cg base pair. Okay, so 2-aminopurine and nitrous acid, it can do the conversion both way. Hydroxylamine can do only one-way conversion. Okay, now acridine orange, what it is? It's an intercalating agent, and intercalating agent results into small or one nucleotide insertion, deletion mutations. Okay, so for that, so for analyzing the table, the only thing you have to remember is what kind of mutation this all agents can incorporate. All right, now let's analyze. So C1 is reversed by 2-aminopurine, nitrous acid, hydroxylamine, not by acridine orange. Okay, now here from not, it is more important who is reversing. Okay, so since these 3 are reversing and they all are causing transition only, that means the mutation caused by C1 is for sure transition. Okay, now C2 is not reversed by any of these agents, means it cannot be transition, it cannot be small insertion or deletion. Okay, now C3, so leave it by blank. So C3 is reversed again by 2-aminopurine and nitrous acid. They are also causing transition. So C3 is also transition. C4 is not reversed by any of these transition producing mutagens, but it is reversed by acridine orange, which causes small insertion or deletions. Means C4 has to be insertion or deletion mutation. Okay. Now from your analysis, you have to look in the table here. Okay. So C1 for sure transition. So C1 transition is in 1. Option 1. Option 2 you can eliminate. Option 3 also you can eliminate. Option 4 also you can eliminate. Okay, so automatically you have got it. Let's check for other one also. So C1 is transition, correct. C2 causes translation or large deletion, that is also true, because C2 is not reversed by any of this. So from this table, we can only identify that it is not transition or not any small insertion or deletion. Can it be transversion? Yes, possible. Can it be a large deletion? Yes, possible. Okay. Now C3 is again producing transition. True. C4 is single base insertion or deletion. Yes, absolutely correct. Okay, so answer will be option 1. Now, again, if you have checked the previous year question papers, the last time question paper also had a question like this. And this question, like if you check last 6 or 7 years paper, no, so at least 5 times this kind of question has come. Where like only difference will be like how many mutagens they have treated or how many stable they have given. All right. So this question is very, very popular. I hope all of you understand how to solve this kind of question. It comes in part C and it is, so since it is popular, so we can say that it is important. Okay. And relatively easy to solve. Only thing you have to remember is what kind of mutation is induced by these different mutagens. Moving on to next question. This is a very interesting question. Okay, and it is somewhat related to your gene mapping also, or the concept of linkage mapping. Okay, now let's see this question. It's a very, very interesting question. So fertilization of gametes containing chromosome with duplications or deletion often results in children with disabilities. What is the probability of a couple where the male is karyotypically normal and the female has a pericentric inversion in heterozygous condition, producing a child with disability if crossing over take place within the pericentric inversion in 26% of the meiotic divisions? In this case, consider that fertilization within a gamete containing chromosome with duplication or deletion will result into disability. So one thing that is mentioned very clearly is that in the female, it is pericentric inversion in heterozygous condition. Okay. Now this is also very important, guys, because अगर homozygous condition में कोई भी inversion है, अगर pericentric है कि paracentric है, homozygous condition में है मतलब क्या हुआ कि दोनों homologous chromosome में same inversion है. So अगर यहाँ पे भी it is DCE, so दूसरे वाले में भी it will be DCE. ओके, तो अगर DC अगर यहाँ पे भी DC है और यहाँ पे भी DC है, so even though it is inverted does not cause any problem in meiosis because alignment तो हो ही जाएगा दोनों के बीच में. Through homology they will align, so there will be no problem or no complication in crossing over if the inversions, whether it is pericentric or paracentric, in homozygous condition. एक बार इस पे भी क्वेश्चन आया था, ठीक है, क्या कॉन्सिक्वेंस होगा, पर उसमें मेंशन था कि वो होमोजाइगस कंडीशन में है. अगर होमोजाइगस कंडीशन में इन्वर्जन है तो कोई भी दिक्कत नहीं होगी, चारों के चारों गैमेट्स विल बी वायबल. ओके, बट यस, सो इसीलिए यहाँ पे मेंशन्ड है कि हेट्रोजाइगस है. अगर हेट्रोजाइगस है देन कॉम्प्लिकेशन विल अराइज. ओके, व्हाट विल बी द कॉम्प्लिकेशन? दैट 50% ऑफ द गैमेट्स विल बी वायबल, 50% विल नॉट. ओके, सो कॉम्प्लिकेशन क्या होगा? 50% वायबल, 50% नॉन वायबल. ओके, नाउ ये नॉन वायबल का मतलब यहां पे दिया हुआ है डिसेबिलिटी. right? So what is it written? Fertilization of gametes containing chromosomes with deletion or duplication will result into children with disabilities. Okay. Now the question comes that if crossing over took place within the pericentric inversion in 26% of the meiotic divisions, then what will be the probability of producing child with disability? ओके, सो इन दिस केस कैसा होगा कि अगर 26% मेयोटिक डिविजन्स हो में इन्वर्जन हुआ है, ठीक है, तो उसका भी 50% वायबल होगा, 50% नॉन वायबल होगा, राइट? सो जो 50% नॉन वायबल होगा, okay, so 50% of 26% that will result into disability. 50% of this 26% they will result into disability. Okay, so क्या होता है? 50% of 26% will be how much? 26 divided by 2%. Okay, so it will be equal to 13%. So here the calculation part is very, very easy. The only thing here is to applying the concept. है ना? तो इसमें बस नंबर्स के साथ करना था. हमको ऑलरेडी पता है कि if there is pericentric inversion in heterozygous condition, then only 50% will be viable, 50% will be non-viable. Okay, so the 50% which is non-viable, they are the one which will result into disability. So 50% of 26% will be 13%. Answer is option 2. So how is it going, guys? Are you finding it interesting? Are you finding the questions relevant? Are you understanding the concepts? Can I have some responses? Okay, Amit. What about others? Doubts are coming. Okay, Amit, please write it down. Please write down your doubts and we'll talk about it in the Zoom class. Okay, Jaya. Excellent. All right. Moving on to the next question. Okay. Now, this question is regarding the numerical aberration in the chromosome, okay, where there is mutation which results into different numbers, okay, difference in the number of chromosomes present. So a species of plant, that is species 1, is diploid, that is 2n equal to 6, with chromosome capital A, capital A, capital B, capital B, capital C, and a related species, that is species 2, is also diploid where 2n equal to 6. Okay, with chromosome PPQQ. The following statements were given by students regarding the chromosome numbers involving this plant species. Okay, so यहाँ पे 5 statements दिए हुए as usual, A, B, C, D, E. The combination of statements with all correct answer is. Okay, तो एक एक करके हम सारे statements देखते हैं. So first statement is talking about auto triploid. ठीक है, इसमें हम इस question को specifically handle करने के लिए it is very important that you understand the terms and the terminologies and meaning of each of these terms, like auto polyploid, allo polyploid, triploid, tetraploid, monoploid, ठीक है, monozomy, nullizomy, trizomy. So ये सारे terms बहुत ज्यादा clear होने जरूरी है. Okay, now let's see the statement. So autotriploid of species 1 will have 12 chromosomes. Now species 1 का क्या scenario है? So this is species 1, this is species 2. Okay, now species 1 में 2n is equal to 6. Okay, so let's check statement 1. So statement 1 is saying autotriploid. Autotriploid, first of all, it is triploid, means there are 3 haploid sets. Auto means self. So the extra haploid set that is added here is of self. Okay, so क्या होगा यहाँ पे 2n plus another n of species 1 only. Okay, so अगर 2n is equal to 6 है तो इसका n equal to कितना होगा 3. Haploid set is 3, means where all chromosomes are present in 2 copies, so that number is 6, means there are 3 different chromosomes are present. Okay, so autotriploid अगर हम species की बात करते हैं तो उसमें क्या होगा 6 plus 3, that is 9 chromosomes will be present and not 12. That means statement A is wrong. Now statement B says allotetraploid of species 1 and 2 will have 16 chromosomes. Now species 2 का देखते हैं क्या दिया हुआ है 2n is equal to 4. So if 2n is equal to 4 means n equal to कितना होगा 2. So haploid set में 2 ही different chromosomes है. Okay. Now allotetraploid. So allotetraploid. Allo means different. Tetraploid. So total होगा 4n. So 4n कैसे बनेगा जब हम 2n1, so statement D अगर देखते हैं तो अगर allotetraploid बनाना है तो क्या होगा 2 of n1 plus 2 of n2. That will come together, then only we will get an allotetraploid. ठीक है, तो अब क्रोमोसोम नंबर अगर देखते हैं तो allotetraploid में कितने क्रोमोसोम्स होंगे. So 2n1 is 6, 2n2 is 4, so it will be equal to 10. यहाँ पे क्या दिया हुआ है 16, so again it is wrong. Now statement C देखते हैं. It is talking about monozomy. Now monozomy is an aneuploidy condition. जहां पे भी suffix zomy लग गया means वहां पे aneuploidy आ गया. Aneuploidy means where we are adding or deleting one or more chromosomes but never the haploid set. Okay, we are not touching the entire set. So C statement says that monozomy of species 1 will generate 5 chromosomes. So monozomy of species 1. Monozomy कैसे होगा 2n minus 1. Where only one chromosome is missing. So 2n1 minus 1 will be monozomy of species 1. So monozomy of species 1 क्या होगा 6 minus 1, that is 5. Now this statement C is correct. Now D says that a double trisomy of 1 will generate 8 chromosomes. अब double trisomy क्या होता है? Double trisomy मतलब first of all trisomy मतलब क्या? Means एक chromosome में like it should have 2 copies of each chromosome, but for one homologous chromosome there are 3 copies present, right? तो उसको हम कैसे लिखते हैं 2n plus 1, that shows trisomy. Now it is double trisomy means यहाँ पे 2 ऐसे homologous chromosomes है जिनका एक extra copy present है. Okay, so 2n plus 1 plus 1. So this is n1 because we are talking about species 1. So 2n1 is equal to 6 plus 1 will be equal to 8. D statement absolutely correct. Now E, as nullisomy species 2 will generate 2 chromosomes. अब nullisomy क्या होता है? Nullisomy means एक situation where both the members of a particular chromosome is missing. है ना? So nullisomy कैसे दिखाते हैं? We show nullisomy as 2n minus 2. So here we are talking about species 2 to 2n2 minus 2 means 4 minus 2. That will be equal to 2. So this statement also is correct. So what are the correct statements? C, D and E. Sorry. Answer is option 3. So as you can see here that this question also is very, very easy. The only thing you have to know is euploidy and aneuploidy, which are the 2 numerical aberrations in chromosome. अगर वो आता है, अगर terms आपके clear है, तो ये question बहुत ज़्यादा आसान question है. All right, let's move on. Another question similar to the previous question where we talked about paracentric inversion. So following diagram shows meiotic pairing in an inversion heterozygote and a point where single crossover has occurred. वेरी वेरी सिमिलर क्वेश्चन, फिर से स्ट्रक्चर दिया हुआ है, यहाँ पे बताया है कि हेट्रोजाइगस कंडीशन में इन्वर्जन है, और अगेन देयर आर स्टेटमेंट्स गिवन रिगार्डिंग द गैमेट्स. You have to identify the correct statements. ठीक है, सो करेक्ट स्टेटमेंट पता करने के लिए सबसे पहले आपको ये पता होना चाहिए कि कौन से टाइप का इन्वर्जन है ये. ओके, सो यहाँ पे कौन सा इन्वर्जन है, जल्दी से बताइए. कम ऑन, I want a response from everyone. What type of inversion is this? The diagram that is showing here. What kind of inversion is this? Paracentric or pericentric? Absolutely correct. Amit, what about others? Yes, what about others? Jaya, Priyadarshini, Neetu, Aisan. Absolutely right, Jaya. Yes, guys, it is pericentric inversion, isn't it? अच्छा, inversion में अगर confusion होता है ना कि paracentric है कि pericentric है, तो उसके लिए बहुत easily ये याद रखो कि pericentric अगर हुआ तो what is the spelling? This is how we write pericentric, okay? तो इसमें से IC निकाल लो. IC will mean include centromere. ठीक है, तो अगर ये याद रखोगे तो कभी भी कंफ्यूजन नहीं होगा कि पैरासेंट्रिक में सेंट्रोमियर आता है कि पेरिसेंट्रिक में सेंट्रोमियर आता है. ओके, सो यस, सो दिस इन्वर्जन, सो इन्वर्जन कौन से सेगमेंट में हुआ है, CD. मींस इट इज अ पेरिसेंट्रिक इन्वर्जन. ओके, सो जैसे ही पता चल गया डायग्राम देख के कि पेरिसेंट्रिक इन्वर्जन है, तो इजी हो गया. अब पता करो ये पेरिसेंट्रिक इन्वर्जन में क्या क्या कॉन्सिक्वेंस होता है. तो इन द सेम वे दैट वी सॉ अबाउट पैरासेंट्रिक इन्वर्जन, सो हियर आल्सो द half of the gametes will be inviable, half of the gametes will be viable. Okay, the gametes which are inviable, that is because of deletion of some genes and duplication of some genes. Okay, but there won't be production of any dicentric chromosome or any acentric chromosome. Okay, so consequence will be chromosome having normal gene sequence, that is true. 50% of the gametes will be like that. Chromosome having inverted gene sequence, that is also true. Other 50% will be like that. Okay, so the chromatids that the gametes which has received the chromatids which has not participated in crossing over. Okay, so in that one of the some of the gametes will have the chromosome in the correct sequence. Okay, A, B, C, D, E, F, G. And some of them will have it in the inverted orientation, but all the genes will be present. Okay, so they both will be viable. See, a dicentric chromosome with deletion and duplication. As I told you, in pericentric inversion, if heterozygous condition, if crossing over happens, dicentric नहीं बनेगा, acentric chromatid भी नहीं बनेगा. Okay, now some of the 50% of the gametes will receive the chromatids which has participated in crossing over, the recombinant chromatids, and they are the one which will be non-viable because of deletion and duplication of the gene. Okay, so they will be non-viable. So which one are the correct statements? Most appropriate for the diagram shown will be A, B, and E. That is option 2. ठीक है, तो ये याद रखने के लिए what you guys can do is you can quickly make a table, okay, and you can write the consequence of paracentric inversion and pericentric inversion. So on the same page you will have a comparative analysis. All right, guys. So I have some extra questions, but we are up with the time here. I request the team to put the question, put the link for the freebies as well as the link for the Zoom class here. So all the students, whoever want to get your doubts cleared by me, like any kind of doubt that you have, you want to ask, please come on the Zoom class. Please click on the link and come on the Zoom class, and we can have a one-on-one discussion there. Okay, I hope to see you all there in the Zoom class. Thank you everyone for joining. Please come to the Zoom class. In just 2 minutes, I will start the Zoom class.